Now, this is a program that was initiated together with a collaborator, Sonia Sharma, and her laboratory at the Jolla Institute of Allergy and Immunology. As many of you aware, the immune checkpoint blockade has really transformed solid tumor therapeutics, in which various therapeutics can be leveraged to reawaken the immune system in order to target tumor cells. Now, as you can imagine, there's tremendous heterogeneity in the response to these various therapeutics, with some individuals having a remarkable response and others having a minimal response to these therapies. In addition, by reawakening the immune system, individuals are prone now to autoimmune adverse events. And so there's been great interest here at Sapient in being able to leverage our tools to understand not only those individuals who are most likely to respond to these therapies, but also to be able to denote and sub-stratify those individuals that are likely to have an autoimmune event in the setting of immune checkpoint blockade-based therapies. To begin with, we were quite excited about understanding particular biomarkers in circulation that read out the PD-1 pathway and may denote those individuals who are more likely to respond to a particular therapeutic in this space relative to folks who are likely to not respond. To begin to initiate this program, we looked at those common genetic variants occurring in the proteins that are central to the PD-1 pathway. And when we look in Sapient's human biological database across the tens of thousands of circulating biomarkers in relation to common genetic variants in the PD-1 pathway, we find that there's over half a dozen molecules, as shown here in red, in circulation that are highly specific and statistically associated with changes in the PD-1 pathway. Now, one of these markers circled in blue here is particularly interesting to us as it appears to be very specific for the actual PD-1 pathway and reads out those individuals in which there's higher versus lower overall PD-1 pathway activity. If we look at this one singular biomarker now in individuals that are undergoing PD-1-based therapy, what we find is relative to pre-treatment, this marker acutely increases in the post-treatment stage. Now, even among these individuals, you can see that there's an underlying heterogeneity in which post-treatment, some individuals have a very high level of this marker, suggesting very high levels of PD-1 activity, whereas other individuals have a more subtle elevation in this one specific marker. If we sub-stratify individuals relative to this single biomarker that's reading out PD-1 total activity, what we find is that this has very important implications for actual response rate. So those individuals that are at the highest quartile for response, meaning that they have the highest level of induction of this PD-1 biomarker, actually are associated with a 50% increase in response to these overall therapeutics relative to those individuals that are in the bottom quartile for response to these actual biomarkers that denote the PD-1 pathway activity. In this way, we can begin to not only identify biomarkers of this immune-related activity, but also to leverage these biomarkers now to sub-stratify individuals according to those who are most likely to respond to a particular therapeutic in this space. Similarly, we can use these techniques to understand markers that denote individuals who are likely to have an adverse event, particularly an adverse autoimmune event in the setting of immune checkpoint blockade. This program was initiated across several human studies, both discovery and validation, as well as preclinical models in which we had autoimmune measures. These biological samples were analyzed using our high-throughput RLC-MS systems with the generation of this non-targeted mass spectrometry data that underwent rigorous biostatistical analysis to identify candidate autoimmune metabolites that are associated with the development of severe immune-related adverse events in the setting of checkpoint blockade. Now, to begin with, we initiated this study looking at a particular clinical trial of one such agent targeting the CTLA-4 pathway. And when we look across these biological samples, again, assaying tens of thousands of these circulating biomarkers as shown on the volcano plot on the left, we can begin to statistically ask, after adjusting for multiplicity, are there specific markers in blood that denote those individuals who are likely to have a severe immune-related adverse event in the setting of immune checkpoint blockade? What we find is that one such molecule, as circled in red, or as denoted in red and circled in blue here, is particularly altered in those individuals that are developing autoimmunity in the setting of these various therapeutics. Now, if we look at the change in this one molecule over time in individuals, what we find is that those individuals who are free from adverse events actually have flat levels of this molecule. Similarly, those individuals that develop grade 1 or 2 mild autoimmune disease, such as mild colitis or mild dermatitis in the setting of these therapeutics, similarly have a very flat level of these markers. Those individuals, however, that develop severe grade 3 or grade 4 autoimmune events, particularly dose-limiting events that can be life-threatening, we find that there's an acute drop in these biomarkers that precede the actual clinical development of autoimmunity. In this way, this singular biomarker may be leveraged to diagnose individuals at a very early time point who may be prone to developing of autoimmune events in the setting, once again, of these immuno-oncology-based therapeutics. Now, this was a discovery that was made in one cohort when we'd be able to thankfully extend it to various other therapies and various other studies of immune checkpoint blockade for solid tumor therapeutics. We can see across 3 independent studies from 3 very different places in the world and very different populations of solid tumors, there's a similar phenotype by which this singular biomarker is decreasing in those individuals that develop severe immune-related adverse events. Across a meta-analysis, we can see the log odds ratio is consistent, denoting or suggesting a universal response to various therapeutics in this space. Now, we can use Sapient's human biological database to look at normal individuals who are free from solid tumors and ask similarly, how does this singular biomarker map onto the development of canonical autoimmune disease over time? What we find is that across more than 10,000 individuals followed for over 10 years, low levels of this marker are associated with increased level of canonical autoimmune disease, whether it be lupus, RA, psoriatic arthritis, or similar disorders, as shown in the Kaplan-Meier plot to the right. Now, we spent quite a bit of time understanding the underlying biology of this single biomarker and how it may be reading out individuals who are prone to developing severe immune-related adverse events in the setting of immuno-oncology agents. What we find across normal individuals in which we have immune cellularity measures is that this one singular biomarker is very much related to circulating levels of a particular subtype of neutrophils, as denoted in the image on the top upper left. Across these individuals in large populations, as would be expected, we find that the biomarker is very much related to circulating neutrophils, in that those individuals that have lower level of the biomarker, as shown on the image on the bottom left, have elevated levels of neutrophils. And we can see through the quartile analysis, as this biomarker level increases, that the level of neutrophils decrease. Now, if we look at these specific subpopulations of neutrophils in individuals undergoing immuno-oncology-based therapeutics, what we find over time, as perhaps would be expected, is that these populations of immune cells are specifically increasing in individuals, particularly those individuals that are developing severe grade 3 or grade 4 autoimmune events in the setting of these therapies. Now, this is a marker, given its underlying conservation, as Tanya alluded to earlier, that we can also begin to evaluate and validate in preclinical systems. We've examined 2 preclinical systems, a humanized CTLA-4 mouse that when upon receiving CTLA-4 and PD-1-based therapeutics develops an autoimmune colitis, as well as the DSS colitis mouse model that many of you are familiar with. To begin with, on the bottom left here, in the humanized CTLA-4 mouse, upon induction with CTLA-4 and PD-1 therapeutics, there's an acute colitis, and similar to as observed in humans, we can see an acute drop in this one single autoimmune-based biomarker. When we look across the spectrum of preclinical samples in the middle image, what we find is the biomarker is very much related to the underlying degree of toxicity, with those preclinical systems having the lowest level of biomarker being associated with the greatest degree of toxicity. Similarly, in the DSS colitis model that has a component of autoimmunity, what we find is with induction of the colitis, there's a similar decrease in overall biomarkers, as is phenotyped in the humans and the CTLA-4 mouse. Now, interestingly enough, these approaches allow us to discover new correlated biomarkers that may denote individuals at high risk. But in this case, we are also understanding biology that has causal relation to actual autoimmune toxicity. So as we've seen in the CTLA-4 mouse, there's an acute drop in this marker that's associated with acute toxicity, and repleting this one biomarker in circulation through IP injection of the molecule actually rescues the underlying toxicity that's seen by the histology score on the bottom left. Similarly, in the DS colitis model, rescuing the levels of this one single molecule results in a rescue of the underlying autoimmune colitis as marked by the histology score. Now, these changes in the actual histology metrics around toxicity are similarly associated with drops in circulating neutrophils upon repletion of the singular biomarker, suggesting a causal relation, once again, between this one unique biomarker, the development of autoimmune events, and neutrophilia.